Look at the black tumors bulging off the side of this catfish. For 10 years, everyone blamed the lake. Pollution, storm runoff, something in the water poisoning the fish. They were wrong. The cancer itself is contagious. It is passing from fish to fish, and the tumors growing in completely different animals carry almost the same genome. That should be impossible. A tumor is normally a dead end. It grows inside one body, and when that body dies, the cancer dies with it. It has nowhere to go. So the real question in this story is not whether these fish are sick. It is how you prove that a tumor has escaped its original body and started spreading like a parasite, instead of each fish simply catching its own cancer. Here is the trick that answers it, and it is pure genome reading. If every fish caught its own melanoma, then each tumor would be built from that fish's own cells. Its genome would look like the host it grew in, plus a handful of new mutations. Line up many fish, and every tumor should hug its own owner. But if one lineage of cancer cells is jumping between fish, the tumors would look like each other across different animals and stop resembling the fish carrying them. So which picture is real? A team led by Emily Curd, Samuel Hart, and Julie Dragon at the University of Vermont sequenced the genomes to find out. Their paper landed in Nature in July 2026. They started with the smallest genome in the cell, the mitochondria. Every cell carries hundreds of these tiny power plants, each with its own little loop of DNA, which makes them easy to read, even in a messy sample. And the tumor mitochondria came back strange. Healthy fish tissue was clean, carrying at most a few odd mutations. Every single tumor carried dozens, 10 times more, and they were the same dozens, showing up tumor after tumor. When they built a family tree from those mitochondrial signatures, the tumors did exactly what a contagious cancer should do. They clustered into their own branch, separate from every fish, and that branch held steady across fish sampled in 2015, 2019, and 2023. Different years, different fish, the same tumor lineage. The mitochondria alone could be a fluke, so they checked the main genome, the nuclear DNA, where almost all our genes live. It told the same story, louder. Across the fish, the tumors shared hundreds of thousands of mutations with each other that their own host tissue did not have. Not dozens, hundreds of thousands. To see how bizarre that is, compare it to human cancer. Researchers looked at hundreds of human melanomas. Almost every mutation in them was unique to a single patient. Line up 16 human melanomas, and you find essentially no mutations shared across all of them, because each person's cancer arose on its own, in its own body. These fish tumors do the opposite. They share their mutations by the hundreds of thousands, which only makes sense if they are copies of one original cancer, passed around. Every other line of evidence pointed the same way. Large chunks of rearranged chromosome, the same in tumor after tumor. Whole chromosome gains and losses that matched other tumors far more than they matched the fish. And when the team hunted for a virus or a microbe that might be doing this, they came up empty. There was no infectious bug hiding in the tumors. The infectious thing was the tumor cells themselves. So this is a cancer that became something like its own species, a cell line that broke out of some original fish long ago and now lives by moving from body to body. That has only ever been documented 3 times in the natural world, which is what makes a fourth so startling. The first is in dogs, a sexually transmitted tumor that spreads between them worldwide, thought to have started in one animal thousands of years ago. It is usually survivable, and it is the oldest known cancer lineage still alive. The second is in Tasmanian devils, a facial tumor they give each other by biting during fights, and this one is a killer. It has swept through wild populations and pushed the species toward collapse, and it has actually arisen more than once. The third is in shellfish. Clams and mussels pass a leukemia-like cancer through seawater, and in some cases it has even jumped from one bivalve species to another. 3 cases, 3 very different animals, all on land or in the sea. Until now, none in a fish and none in freshwater. Which raises the obvious question: how does a cancer get from one fish to another in a lake? Honestly, nobody has caught it in the act yet. The leading guess is spawning. Brown bullhead crowd together in the shallows to breed, pressed body to body. And these are scaleless, bottom-hugging fish, so living tumor cells might rub off and take hold on a mate. It could also travel through the water or the sediment. Notably, no young fish have the disease, which fits a cancer that spreads during breeding, or one that needs a long time to take. And there is a lot still open. The team does not know how old this lineage is, or which fish it first came from. The nuclear DNA even hints it may have started outside this lake, in fish further south, though that is not pinned down. They do not know how hard it hits the population over time, or how far it has already spread. Those are the next questions, not settled answers. One thing they can say clearly, because people always ask: there is no known risk to humans, and no known risk to the drinking water this lake supplies to well over 100,000 people. This cancer moves between bullhead catfish. It is not built to cross into us. And it does not erase the pollution story completely. The lake really did carry elevated arsenic and other metals, and that kind of stress may make a fish easier to infect. What changed is the cause of the tumors themselves. They are not many separate pollution cancers. They are one contagious cancer, and now we can read it in the genome.